In this example of complete dominance, a mother and her child have different alleles for the genes involved in one type of albinism. The mother is heterozygous, while the child is homozygous recessive. Both the mother and father of this child are heterozygous, so their gametes have an equal chance of carrying a dominant or recessive allele. Their children have a 25% chance of inheriting both recessive alleles and having albinism. With complete dominance, individuals who inherit either one or two dominant alleles are normally pigmented. How does complete dominance work at the molecular level? In this example of albinism, the dominant allele codes for an enzyme called tyrosinase, which converts the amino acid tyrosine into the brown pigment melanin. With either one or two copies of the dominant allele, enough tyrosinase is produced to generate normal amounts of melanin. Homozygous recessive individuals lack a functioning tyrosinase enzyme and produce no pigmentation. Complete dominance is not the only possibility for a heterozygous genotype. Sickle cell hemoglobin provides an example of a pattern of inheritance called incomplete dominance. A typical red blood cell carries between 200 and 300 million molecules of hemoglobin, the molecule that binds and carries oxygen molecules. A red blood cell filled with normal hemoglobin, designated HbA-HbA, is a biconcave disc. In sickle cell anemia, designated HbS-HbS, most red blood cells appear normal when the oxygen content of the blood is high. However, as oxygen levels drop, such as when red blood cells deliver their oxygen to the body's tissues, the cells become sickle-shaped and tend to clump together, clogging capillaries and depriving tissues of oxygen. The shape results from the abnormal hemoglobin molecules sticking together in long polymers, which does not occur with normal hemoglobin. People with sickle cell anemia usually die at a young age. Heterozygous individuals are said to have the sickle cell trait. The normal allele shows incomplete dominance, meaning that the expression of the heterozygous condition lies somewhere between the two homozygous states. People with sickle cell trait are generally healthy, but sickling and clumping of red cells may occur if there's a prolonged drop in the oxygen content of the blood, as might happen during travel at high elevations. Because normal hemoglobin molecules reside in the same red blood cell as abnormal hemoglobin, polymerization does not occur very readily. If two individuals with the sickle cell trait reproduce, their children will have a 25% chance of having sickle cell anemia, a 50% chance of having sickle cell trait, and a 25% chance of having normal red blood cells. This pattern is typical of incomplete dominance. In another pattern of inheritance called codominance, the effects of both alleles are separately apparent in a heterozygote. For example, the AB blood type arises from the presence of a type A polysaccharide and a type B polysaccharide. Two alleles of the same gene, IA and IB, code for different enzymes, which produce the A and B polysaccharides respectively. Populations have the IA and IB alleles, as well as a third allele called i, which doesn't code for an enzyme. People with a homozygous recessive i genotype have type O blood, which lacks the A and B polysaccharides. Blood type A results from a genotype of IA-IA or IA-i, and blood type B results from genotype IB-IB or IB-i. Note that while the IA and IB alleles are codominant to each other, each is completely dominant to the recessive i allele. Knowing the patterns of inheritance of blood type alleles, we can make predictions about the outcome of a genetic cross. If a man with type A blood of the genotype IA-i and a woman with type B blood of the genotype IB-i had children, those children would have an equal chance of having blood type AB, A, B, or O.